Hi, my name is Blanco Osorio, and my group will be presenting the generation of monoclonal antibodies, immunoprecipitation, and immunoblotting. Monoclonal antibodies are lab-produced proteins cloned from a single plasma B cell. They are used to enhance, restore, mimic, and inhibit immune function. They're mostly used for the delivery of therapeutic agents. There are 4 types of MABs. Mouse and chimeric MABs cause allergic reactions in humans, so only humanized and human MABs are accepted for clinical use. The most common process for producing MABs is immortalizing B cells. A subject is injected with an antigen carrying the desired epitope, and in response, the antibodies are produced. B cells that produce those antibodies are removed and fused with an immortal cell to produce hybridoma cells, and those hybridoma cells produce an endless supply of MABs. Specificity is the #1 parameter. Without it, MABs could bind to the wrong region or mutate. The second is ensuring immortalized cells are mutated so they're unable to produce their own antibodies. The third parameter is the purification step, and here antibodies from fused cells are screened by being placed into a hat culture where only hybridoma cells are able to survive. MABs produce information for diagnostic treatment, passive immunity, identification of cells, and even purify concentrations of molecules and mixtures. Humanized MABs have been used in the treatment of cancer, lupus, transplant rejection, and most recently, COVID. Engineers should consider its expensive production, long production time, lifetime of about 20 days in the body, and its large production yield.
Immunoprecipitation, or IP, is a technique that is used to study and target a specific protein or protein complexes present in small volumes in biological tissues. It consists of 3 parts: incubation, separation, and analysis. There are 2 different types of IP, immunoprecipitation and co-immunoprecipitation. IP targets a specific protein. Here we add an antibody that targets this protein, and we add beads that will bind to it, and we pull it down by centrifugation. In co-immunoprecipitation, we do the same procedure, and we're pulling down an entire complex as opposed to just one protein. Some things to consider when doing an IP are choosing the right lysis buffer, considering pH, specificity of our antibody, and washing and eluting. Once we have our purified sample, we can identify proteins by mass spec and quantify them by using Western blots or analyze them using PCR. With IP, we're able to study unknown proteins, and it offers faster incubation times over other methods. We're also able to keep the proteins in their native conformation. IP is widely used in science. One IP assay called luciferase immunoprecipitation system is used to identify and quantify antibodies against diseases such as COVID-19 in previously infected people and measure autoantibodies in several human autoimmune diseases, including type 1 diabetes. IP is a great assay because protein that is present in small volumes in large samples can be obtained with high sensitivity and high signal-to-noise ratio. IP is a reproducible assay, relatively inexpensive, and the process can be automated and completed in 1.5 hours. Additionally, there's a huge variety of IP kits in the market to target different proteins.
Immunoblotting, also known as Western blot, is another biomolecular lab technique just like immunoprecipitation. It is used to identify a specific protein within a heterogeneous sample. One direct application of this is diagnosing disease by detecting proteins associated with pathogen. Another application is verifying the translation of a cloned gene in molecular cloning. This technique takes advantage of antigen-antibody specificity. We use a primary antibody to detect the protein of interest, then a secondary antibody to detect the primary antibody. The secondary antibody is a fusion protein, meaning that it is connected to an enzyme to produce a detectable signal such as luminescence. Lastly, immunoblotting uses gel electrophoresis, which is a way to separate different protein by mass, since lighter protein can travel further through a gel than heavier one. The general procedure goes like this. First, we lyse the cell with detergent. Next, we separate protein by size using SDS-PAGE, which is a type of gel electrophoresis. Then the protein in the gel is transferred to a nitrocellulose membrane. After that, we add the primary antibody. Then wash away any unbounded primary antibody. And lastly, we add the secondary antibody. This is what the output of a Western blot looks like. Each vertical lane is a different sample being analyzed, and each band is the protein detected by the primary antibody. The darker a band is, the higher the concentration of the detected protein. And larger protein can't travel as far through the gel and are located near the top.